I'm a second-year PhD student in James Sands' lab, and I'm here to give the membrane perspective on Mycoplasma and SYN3, and also why there's such exciting minimal organisms from the membrane perspective. So to jump in quickly, we know that membranes are essential to life, and we know that they have highly conserved properties. Cell membranes are semi-permeable. They have to allow in some, but not all things. They have to be robust to the external environment, and they also have to be responsive to changes in the external environment. And this means they have to be dynamic. Cell membranes have no equilibrium state. They're always remodeling their composition. Finally, we take this for granted, but cell membranes are lipid-based, and there are a number of reasons for this. The thing is, though, we still don't understand cell membranes. We still don't understand why specific lipids are chosen to be or not be included in the membrane, and we don't understand the regulatory aspects of membrane remodeling. So now I'm going to give a very brief overview of what lipids are for anyone who hasn't encountered them since undergraduate classes. So most phospholipids have a polar head group and then non-polar fatty acid chains, which can either be saturated or unsaturated with a double bond. And there's a massive diversity of lipids, both within class and species. So class diversity has to do with the head group, and there are a number of different head groups with various properties. And then you also have species diversity within a class, and this has to do with the number of saturations, so either singly unsaturated, fully saturated, or polyunsaturated. It also has to do with the number of chains. 2 is normal for most phospholipids, but some like cardiolipin can have 4, or lysophospholipids can just have 1. And it also has to do with the number of carbons in each chain. So you can have a number of different variables that can be varied, and this leads to a massive amount of diversity. And the plasma membrane is a fluid mosaic, if you think back to these undergraduate classes. And what that means is most of the membrane is composed of lipids. You have a class of lipids called sterols in the membrane of Mycoplasma and SYN3 that modulate the fluidity of the membrane. And then you have peripheral and integral membrane proteins. And the composition of the lipid determines the membrane properties, which is very important for membrane protein functionality. So it determines membrane thickness, the local and global charge of the membrane, and also the fluidity, or how tightly or loosely packed lipids are in the membrane. And when the external environment changes, the composition of the membrane changes so that the microenvironment around the proteins remains constant. So the question is, why does life need so many lipids? We know that life has selected for very complex membranes, and the complexity of these membranes makes it difficult to understand what's going on. So that's when we turn to mycoides and SYN3 as simple model membrane systems. And there are a number of reasons these are excellent model membrane systems. They have some of the smallest known genomes. I don't need to say that to this audience. They have a single plasma membrane with no cell wall, which means it's easy to directly probe the membrane. They're mammalian parasites, so the composition of their membrane mimics that of mammalian rather than prokaryotic cells. So we can make some interesting claims potentially about mammalian membranes while studying this very simple organism. Now they have a lack of lipid synthesis capacity. They can't make their own fatty acids, and this means we have more control over their membrane than we would in another organism. And finally, there's a synthetic analog. So we can look what happens in the already simple mycoides, and then look at what happens in the even simpler SYN3. So mycoplasma have a simpler membrane than most other cells, but when they're grown on a serum diet, they still have a fairly complex membrane. So this data is mass spec lipidomic data that was gathered by my colleague, Natasha Safronova. And the colors show the different classes of lipid. So PCs, PGs, cardiolipins, sphingomyelins, and then cholesterol. And then within a class, you have the breakdown of the different species. So within the class of cardiolipin, you have a number of different cardiolipins with various lengths and saturations within their lipid chains. So mycoplasma on FBS, we might say it's simpler than other organisms, but it still has roughly 100 lipid species in its membrane. So the question is, can we push that simpler? And Natasha spent her PhD developing a system where you can feed mycoplasma and SYN3 defined lipid diets. So instead of FBS, which has undefined lipids in it, you instead use a combination of a lipid loader and specific lipids, and you can really push the complexity of the membrane much, much simpler based on the lipids you feed it. So this was a major breakthrough and allows us unprecedented control over the composition of the membrane in mycoides and SYN3. The thing is, though, even though these are simpler systems, mycoplasma in SYN3 still retain some remodeling capability based on the phospholipases present in the organisms. So the phospholipase we're interested in are PLA1, which cleaves an acyl chain in the R1 position, PLA2, which cleaves the acyl chain in the R2 position. Phospholipase C is a family of phospholipases that is generally involved in making molecules for small molecule signaling, so we care less about that. And phospholipase D, which cleaves the head group after the phosphate and can be used to prepare PG to be remodeled into cardiolipin. So what does that look like when you feed the organism, let's say, POPG? Well, the PLA1 and PLA2s can cleave the acyl chains, and then the PLD can cleave the head group. And once the organism has access to free fatty acids or free acyl chains, as well as the modified head group, it can synthesize saturated or unsaturated lipids based on the access of the fatty acids, as well as cardiolipin. So even with a very simple diet, the organism can still remodel the lipids, which makes it more complex than we could want. So my question was, can I push it even simpler? And that's why instead of using ester lipids, I switched to synthetic ether lipids. You'll notice they have a different bond linking the acyl chain to the head group. And what this means is that phospholipase PLA1 and PLA2 cannot actually cleave the acyl chains at those positions. So the organism doesn't have access to free fatty acids when fed ether lipids. And so I believe I've pushed the membrane of mycoplasma in SYN3 to their minimal state, a minimal membrane for minimal cells. So on mycoplasma grown on just cholesterol and doubly saturated diether PC, singly unsaturated, singly saturated PC, and doubly unsaturated PC, I have living mycoplasma with only 4 lipids comprising over 99% of their cell membrane. This is the simplest reported membrane, and what's even cooler is that it also works for SYN3. So we have living SYN3 that only have 4 different lipid species comprising over 99% of their membrane. And what's very exciting when you push the system this simple and this static is you now have a platform where you can specifically reintroduce complexity. And specifically, now that we have this minimal static platform, I can reintroduce specific ester lipids to the diet, and their remodeling can be studied. And their remodeling can be looked at both in how that affects the biophysical properties of the cell, but also how that affects the regulatory machinery in terms of genes and proteins active in the organism. So what we can do is we can combine changes to the diet with transcriptomic analysis to look at specific genes that are up or down regulated when specific lipids are added or removed from the diet. So this is just an example of data because this is still preliminary work, but this is comparing cells grown on diether PGs to ester PGs. And this is in mycoplasma rather than SYN3, so some of these genes may not be present in SYN3, but what you can see is multi-log fold up or down regulation of specific genes when you change the diet. And what's very exciting about this is you can combine these transcriptomic analyses, proteomic analyses, and predictive structural algorithms to identify membrane proteins of interest. And that's either, as of now, unidentified phospholipases, but also specific lipid transporters that are currently unannotated in mycoplasma or SYN3. Now, once we have the hit list of proteins of interest, the next step is to express those proteins in vitro and also downregulate them in vivo or otherwise modify their functionality in vivo to validate their function and actually get closer to knowing what every protein and every gene in SYN3 is capable of doing. And finally, away from identifying proteins and genes, because we have such a simple membrane system, we see compromises in the fitness of the cell, and we can look at what happens when we modulate both the specific composition of the membrane as well as the complexity of the membrane, and then what happens to both the biophysical properties of the cell as well as the overall cell fitness. So thank you all for listening, and I'm open to any questions.
I am dazzled, Isaac. I really am. You guys have gone a long, long way with this system. So it would seem if we had, if we were to reproduce your media here and then do a transposon bomb, or send you the protocols for it to do a transposon bombardment that might help identify that some genes that might have been essential previously in SYN3 would now be non-essential. Wouldn't you expect that?
I think we would. And that would be really interesting to look at. It's also, I'm really curious in these 7 genes that differ between SYN 3.0 and 3.A,
Mm-hmm.
because if some of them, I've talked with James, who's actually speaking next, as well as with David Bianchi about this, but a couple of those I'm very, very interested in, and I think they might have something to do with remodeling or controlling ester lipids. And so the question is, are those genes necessary to regain control of organism division if you feed them an ether diet rather than an ester diet? So both for those specific genes and more in general, I think some genes may no longer be necessary because you can no longer cleave these acyl chains.
How does changing the lipid diet affect growth rate?
It plummets. And now for a lot of Natasha's diets, you'll see a decrease in growth rate for a couple of passages. And then once the cells are adapted to the new diet, depending on the diet, you can sometimes recover growth rate and you sometimes cannot. But generally, you fairly can. When you push them to these very simple diether diets, the growth rate is extremely compromised and remains that way.
How give me minutes, please. Or a
We don't,
guess.
a guess is, so it's a linear growth rate. It never hits exponential.
Mm-hmm.
And order of magnitude 4 to 8 hours, I would say.
Okay. On the JCVI website, which you have access to, Kim Wise has his video of exactly how he measures growth rate, which is very effective.
Yes. Yeah, I've had more success for some diets than for others.
Mm-hmm.
So part of why I've been thinking about this as reintroducing membrane complexity is when you re-add specifically free fatty acids, now the cells can synthesize PG and cardiolipin. So you see an immediate boost in fitness, but they also are forced to incorporate between 20 and 40 mole percent of their total lipidome remains the diether PC. And so depending on if it's saturated or unsaturated, you can really push the lipidome in a specific direction while recovering the growth rate and recovering sort of normal cell functionality. And then you can use systems like optical density measurements. So with the increase of a little bit of complexity, then we can get better growth rates and the cells behave more properly like cells. I've had a lot of trouble getting any sort of growth rate for these very, very simple diets because the cells are essentially just hanging on as best as they can.
So, other questions, folks?
John, there's I think James has a question.
Beautiful talk, Isaac. I wonder if you could please remind me, is cardiolipin synthase able to synthesize cardiolipin from the diether lipids, or are those cells likely do not have cardiolipin in their membranes?
So what the cells are able to synthesize is if they have access to free fatty acids, they're able to synthesize PG. And if they have access to PG, they're able to synthesize cardiolipin from that PG, provided that it's not doubly saturated PG. So if you either feed the cells free fatty acids or ester phospholipids are capable of cleaving acyl chains from, then you see cardiolipin. For my diether diets, you do not see cardiolipin, except interestingly, if you feed the cells diether POPG, and then the phospholipase D enzyme is still present, And so you'll actually see quad ether cardiolipin synthesized from diether POPG.
Thank you very much.
Troy, you're up.
Thank you for the talk, Isaac. I just had a quick, can you hear me?
Yep, I can hear you.
Okay, I didn't see my mic moving, so just wanted to check. Just had a quick question. I think if I understood it, you said you saw changes in some of the expression based on the transcriptomics for some of the genes. Were all of those genes either lipid associated or uncharacterized genes? Kind of what I'm asking is, did you see, like, let's say a gene that's implicated in, let's say, nucleotide metabolism also having a change in its expression because of the diet? So kind of a lipid impact of the lipid metabolism on other aspects of the cell?
Yes, the short answer is yes.
Okay.
And actually one of the problems I've been having with the transcriptomic data set, and part of why talking to David has been so incredibly helpful, is essentially it tells you what's going on with every single gene. And so if you don't know which genes you're looking out for, it's easy to look at the very significant outliers, but there's still a lot of significant changes happening. And if you don't know what genes to look out for specifically, it's kind of like pulling a needle out of a haystack. So if you, or if anyone wants to email me or write in the chat specific genes or specific conditions you're interested in, I can then look for how the regulation of those genes are changed in my transcriptomic databases.
Okay, thank you. Appreciate it, and good talk.
There is
Thank
a
you.
question. There's also a question in the chat. What are the components that are needed for defined media beyond the lipids from serum?
So Kim Weiss actually made a completely defined media. And in conjunction with a defined lipid diet, we haven't yet been able to recreate the fully defined media. So the way I'm defining defined media here is that we know that the only lipid sources are the ones we include. Whereas carbon sources and other sources, we don't need to be defined in this definition. So I'm focusing just on defined lipid sources, but I know Kim does have a completely defined media.
Yeah, and Kim is in Croatia today and not able to join us, but he will be, even though he's retired, he still seems to be, deeply involved in the whole process. Other questions? I see a hand up from someone.
Zane?
I just wanted to throw in that can you hear me first of all? Sorry, John.
Yeah.
Yep.
The current defined medium composition is in the Cell paper that Zane Schulten's group published with JCVI this past winter. So for anyone who wants to look at that, there's a full components list published in that Cell paper.
And I will say that it is published, but putting this together is difficult. And it's a lot of measuring small amounts and making sure that your reagents are in good shape. But it's doable. James Sands and Isaac, I want to try to put you in touch. So Bernard Paulson's group is looking at some things that seem to dovetail nicely with what you're doing. I don't see any of them in the people who are listening right now. I could be wrong about that, but I'm going to try to set up that maybe a conference call with the all of us to think about how this might work out together.
That would be fantastic.